Glycolysis. Before to discuss glycolysis, let's clear the basic background of the glycolysis. We know metabolism. Metabolism or metabolic activities is the sum of all reactions which take place in the living bodies. Metabolism may be anabolism or catabolism. Anabolism is actually constructive process or constructive metabolic reaction in which food is prepared. And catabolism is actually destructive process in which food is broken down to release energy. Its example is photosynthesis, and the example of the catabolic reaction is a respiration. In photosynthesis, food is prepared from simpler inorganic substances. As we know, food is organic, and then that food is broken down in the process of respiration to release energy. So photosynthesis is constructive and respiration is destructive process. Respiration cannot be completed in a single step. It is multi-step process. The first step of the respiration, I'm talking about the aerobic respiration, is the glycolysis, which we are focusing here. The second is a short reaction, and it is known as link reaction. The third step of the respiration is Krebs cycle. And the fourth and final reaction or step of the respiration is an electron transport chain. So we are going to discuss glycolysis in two terms. First, let's come to discuss the meaning of the glycolysis. The word glycolysis is actually the combined form of two Greek words. One is glyco and other is lysis. The word glyco is actually used for glucose, and lysis means splitting or breakdown. So the breakdown of glucose into two pyruvate molecules to release less amount of energy in the presence of enzymes is known as a glycolysis. We know in the glycolysis only 2 ATP are released, which is actually the energy released in this process. Glucose is broken down into pyruvate molecule. Glucose contains 6 carbon, and pyruvate 2 molecules. Both of the molecules contain 3 carbon. Glycolysis was discovered by 3 scientists. One is Emden, the second one is Myrouf, and Barnes in 1897. The cell membrane of a cell is permeable to glucose, so glucose, when entered to the cell, may undergo glycolysis or other metabolic pathways. The glycolysis process may be aerobic or anaerobic. So, on the basis of availability of oxygen, glycolysis is divided into 2 types: aerobic glycolysis and anaerobic glycolysis. Aerobic glycolysis occur in the presence of oxygen. Anaerobic glycolysis do not need any oxygen. Both of the glycolysis, aerobic and anaerobic, are same in their all aspects, such that enzymes, their products, and their energy released. But here little bit differences are present, which I'm going to discuss. In aerobic glycolysis, 2 ATP are consumed. In anaerobic glycolysis, also 2 ATP are consumed. In aerobic glycolysis, 4 ATP are prepared, and it is same for the anaerobic glycolysis. 2 ATP are consumed and 4 are prepared, so 4 minus 2, net gain of ATP we get in the process of glycolysis is actually 2. We get 2 ATPs. In the aerobic glycolysis, 2 NADH are produced, which are actually coenzymes which take hydrogen to the electron transport chain. And in this also 2 NADH are produced. But it should be noted that in the aerobic glycolysis, these 2 NADH2 give 6 ATPs in electron transport chain. But in anaerobic glycolysis, there is no electron transport chain, so therefore these NADH2 cannot give ATP molecules. These NADH2 are used to convert these pyruvate molecules into alcohol or lactic acid. So these NADH2 are used to convert pyruvate molecules into alcohol or lactic acid. So the question is there that where the glycolysis occur? Glycolysis occur in the cytoplasm of the cell. It occurs in all cells. It do not need any mitochondria, so it is common to all the living organism in all cells. Glycolysis is completed in 11 and 10 steps, in which total 10 enzymes work. It is completed in 3 phases. The first phase is known as preparatory phase. Preparatory phase. It is also known as energy utilizing phase because in this phase 2 ATP are used by the kinase enzyme to attach phosphate to glucose molecule. The second phase is known as splitting phase. In this phase, the fructose 1,6 bisphosphate is broken down into 2 molecules of 3 carbon. And the third phase is known as oxidative phase or energy generative phase. In this phase, 4 ATP molecules are prepared. So now, dear, we are going to discuss the complete description to the glycolysis. When glucose molecule is absorbed by the cell in the cytoplasm of the cell, this glucose molecule converts into glucose 6-phosphate in the presence of enzyme known as hexokinase. Kinase enzyme deal with energy, so it work in the presence of magnesium plus iron. In the first step, as you see, the glucose contain phosphate and carbon number 6. So this phosphate actually come from ATP, and ATP get converted into ADP. In the second step, glucose 6-phosphate is converted into its isomer fructose 6-phosphate. This step is carried out by an enzyme known as phosphoglucoisomerase. In the third step, fructose 6-phosphate is converted into fructose 1,6 bisphosphate, mean 2-phosphate. 1 phosphate is attached on carbon number 1 of the fructose, and the second phosphate is attached on carbon number 6 of the fructose. Here the same enzyme works as in the step number 1, kinase, in the presence of magnesium ion. But this kinase is actually known as phosphokinase or phosphofructokinase. In this step, as you see that another phosphate comes and attached now on carbon number 1 of the fructose. So where from this phosphate come? This phosphate also comes from ATP. So as a result, ATP is converted into ADP. Now, up to this is known as the first phase, or this is known as preparatory phase. This is also known as energy utilizing phase because in this the energy of 2 ATP, one in the first and second and third step, are used, and phosphate is attached to either glucose or fructose. In the next step, which is actually step number 4, The fructose 1,6 bisphosphate is splitting down into DHAP, dihydroxyacetone phosphate, and G3P, glyceraldehyde 3 phosphate. Both DHAP and G3P are isomers of each other, so these can interconvert into each other in the presence of an enzyme known as triophosphate isomerase enzyme. So DHAP converts into G3P, and then G3P molecule goes under the reaction. Now G3P molecule is converted into 1,3-bisphosphoglycerate, in which on carbon number 1 and 3 2 phosphate or bisphosphate are attached. This is done by an enzyme known as glyceraldehyde 3 phosphodehydrogenase. Because hydrogen are removed from G3P molecule and these hydrogen are attached to NAD, so 2 NAD are converted into 2 NADH. This is very important step. Another one phosphate is attached to carbon number 3, but here another phosphate is also attached in carbon number 1. Where from this phosphate come? This phosphate actually come from inorganic sources and get attached on carbon number 1 of the G3P molecule, which convert into 1,3-bisphosphoglycerate. This 1,3-diphosphoglycerate molecule is converted into 3-phosphoglycerate molecule. As we know, 2 G3P molecule are present because this also converted into this. So 2 1,3-bisphosphoglycerate, and these are also 2. This is done by an enzyme known as kinase. Here again I am discussing kinase enzyme, so something related to the energy occur in this step. What occur? Actually, ADP is converted into ATP, 2 ADP converted into 2 ATP. So in this step, this is actually step number 7. Now this 3-phosphoglycerate molecule is converted into 2-phosphoglycerate in the presence of enzyme known as phosphoglyceromutase. It works in the presence of magnesium plus ions. Now these are actually 2 molecules. Now 2-phosphoglycerate molecule is converted into phosphenol pyruvate molecules, 2 phosphenol pyruvate molecules, and 1 water molecule is eliminated in this step from 1 G3P and another from another G3P, so 2 H2O molecules in the presence of enzyme known as enolase. Now these phosphonol pyruvate are finally converted into 2-pyruvate molecule. These 2 pyruvic acid molecules is actually the result of the glycolysis in which glucose is broken down into 2 pyruvate molecules. In the last step, 2 ADP are converted, and this phosphate, last one phosphate, in 1G3 or last in other G3P molecules. So these are attached to ADP to make 2 ATPs. So in the step number 10th, 2 ATP are prepared. And here the enzyme work, which is known as kinase, it work on pyruvate molecules. So it is known as pyruvate kinase in the presence of magnesium plus ion. Now this pyruvate molecule either go to the Krebs cycle and electron transport chain in aerobic respiration, or it may be used to make alcohol or lactic acid in the presence of a NADH2 in anaerobic glycolysis. This is actually from here, this is actually the energy generating phase. And this is splitting phase in which fructose 1,6 bisphosphate is broken down into 2 molecules in the presence of an enzyme known as aldolase. So this is actually the stepwise description to the glycolysis. If you like, please subscribe our channel and keep watching the videos. Thank you.